So in the team, we are actually working on trying to find mechanisms for synapse formation and synapse regulation. And to do that, we need to use some specific imaging techniques, because as you know, synapses are very tiny structure. They are 1 micrometer big, 300 nanometer large. So to look in details at the organization of the synapse and of the synaptic molecules, we turn to a single molecule localization microscopy. And you can see on this example the same dendrite on wide field standard fluorescence microscopy. And here down, it's on using a single molecule localization microscopy. You can see, we can see much better the localization of the yellow molecules in relation to the blue synaptic marker. But we can also see molecules that are at the periphery of the synapse, or also the organization within the synapse of the receptor. So why are we using this approach? It's because we believe it's very important to understand how the synapse is organized, and especially how receptors that receive the information from the release of neurotransmitter from presynaptic site, how these receptors are organized in the synapse, because there is a very direct correlation between the number of receptors you find within the synapse and the efficacy of this synapse. So more you have receptor in the synapse, more the synapse is efficient. And so you can see on this image these receptors in green on a hippocampal neuron that are actually not evenly distributed at the surface of the neurons, but they form these aggregates. We call them cluster, and that's what I schematized here on this drawing. And they are properly, facing the presynaptic button that contains the synaptic vesicles that release the neurotransmitter. So these receptors, they accumulate at this site thanks to some protein that we call scaffolding protein that are represented in blue here. And these proteins, they anchor the receptor to the cytoskeleton. So they are really making the bridge between the receptor and the cytoskeleton. So actually, these these receptors at the synapse, they are not immobile. They move in the membrane, and they move thanks to lateral diffusion. And they can enter the synapse as well as exit the synapse by lateral diffusion. And by this way, they can be internalized, and either they are recycled back to the membrane or they are rapidly degraded. So we know that with lateral diffusion and this mechanism that we call diffusion capture by the scaffold is really one of the first cellular mechanisms to really control this number of receptors at the synapse. And this is very important in particular, for instance, for synaptic plasticity. So all these processes where the synapse respond to a change of activity. And indeed, you can put more receptor in the synapse, or you can put less receptor in the synapse. And this is, roughly correspond to a potentiation of the synapse or a depression of this synapse. And this can be correlated with a change in the rapid diffusion of the receptor at the synapse. So here I represented a trajectory that moves, actually explore a very large surface of the membrane. And this receptor is extrasynaptic most of the time, while here you can see a more restricted trajectory, which happen at the synapse. So if you want to increase the number of receptors at the synapse, you can see that the receptor become even more confined. So it's exploring a smaller surface area, and that's the way it is captured at the synapse and kept them there. While if you want to depress the synapse, you have to increase the mobility of this receptor, and the receptor escape the synapse. So that's exactly what we are looking in the team, it's a correlation between the diffusion of the receptor, and we are focusing mainly on the GABAergic inhibitory synapse, which is the main inhibitory synapse in the hippocampus and in the brain. And we correlate these diffusion changes to changes in receptor number. And so we are using these techniques. So to look at the diffusion, we are using single particle tracking with quantum dots. So I will go more in details in the technique, of course, for on living cells, of course, and for studying the relationship with a change in number of receptor, we are using also another approach of single molecule localization microscopy, which is the storm palm imaging approach. So as you can see on this image, the receptor, for a long time, we thought that the receptors, they were also homogeneous in the synapse, as you can see on this typical epifluorescence image. But now we know that using storm palm imaging, they are not homogeneous. They actually form what we call nanodomains. So, and you have 2 nanodomains for GABA receptors, so we can quantify this data, and they are represented here on this image. You can see 2 nanodomains of receptor per synapse. So why we think it's important? It's because they are properly aligned in front of the releasing site for the neurotransmitter. So they are really positioned in front of the neurotransmitter release. All right, so concerning the 2 techniques I'm going to talk about today, I'm going to start by talking about the quantum dot-based single particle tracking approach. So here is an example. I hope able to see it now. Okay. Let's see if the movie plays. And no, it doesn't play. Of course, it's perfect. No, it doesn't want. Okay. Okay, so it was supposed to be a movie where you could see in red the synapse, and in white the quantum dot, which is a fluorescent probe that can be bound to a receptor thanks to an antibody. And so using this probe, you can actually follow the mobility of the receptor at the surface of the neuron. And on this movie, let's see if it works. Oh my God, nothing works. It's terrible. Okay. Oh my God. Okay, so that's too bad. I cannot show you any movie. That's terrible. Anyway, so we are supposed to see the white dot moving and exchanging from one synapse to the other. Anyway, so concerning the protocol we are using, we actually wrote a protocol a long time ago, Nature protocol, and that you can have access to. And actually, there is no much changes that have been occurring during over time on this protocol. So we are working mostly on hippocampal cultures that we prepare from E18 rat embryos that we extract the hippocampus from the brain of these embryos, and we plate the neurons on glass cover slips, and we culture these neurons for 2 to 3 weeks when they get mature, actually. And then we work with this glass cover slip to actually stain the receptor of interest, so the GABA A receptor, and we use a specific antibodies that are going to recognize extracellular epitopes for the receptor. For the main subunits we are looking are alpha-1 and gamma-2, the ones that you can find at the most important inhibitory synapses. So to do quantum dot based single particle tracking, since this is a single molecule localization microscopy technique, you need to have inverted fluorescence microscope equipped with a very good objective to be able to capture, you know, a very dim signal. So most of the time we are using 63x or 100x objective with a high numerical aperture, which means it can collect a maximum of photon. So you want to collect a maximum of light from your sample. Here you can see 4 quantum dots on this image, and without such equipment you couldn't see them. Also, you need to do quantum dot single particle tracking. You need a sensitive camera, and we are using electron multiplying camera cool device. So it's a camera that can actually sense a single photon and that cool down, so it limits the noise also on your sample, so you can have a good signal to noise ratio. So the quantum dots, I will explain you what it is exactly, but they are very bright dyes, and therefore you don't need a laser to shine them and to get their fluorescence. A mercury lamp or a xenon lamp or a fiber or a simple fiber is enough actually to collect their and to stimulate and to collect their fluorescence. So here is a timeline of a typical experiment. So usually we are using a 2 weeks old culture, and that we can transfect them using a lipotransfection with a synaptic marker. And for instance, you can use a Jeffrey finger GFP with a construct that can encode a nanobody coupled to GFP. And this nanobody is going to stain endogenous Jeffrin, so the scaffolding protein for the receptor, which is a good marker of inhibitory synapses. But you can do that also for excitatory synapses using PSD95, which is also the scaffolding protein for excitatory synapses. So concerning inhibitory synapses, so we use this marker, and the neurons go back to the incubator, and we are waiting for 1 more week that the Jeffrey finger GFP get expressed and label the inhibitory synapse. Then we take out the cover slip that contains the transfected neurons from the incubator, and we do an immunocytochemistry using an antibody that recognizes a specific epitope for the receptor of interest. And this antibody should recognize an extracellular epitope because you are using living neurons, of course. So it doesn't have access to the intracellular receptor. It's access only extracellular receptor, and the quantum dot is coupled to this antibody. Then you acquire the movie, and so the movie, as you can see here, there are many quantum dots on this movie. Usually we do 1200 frames, so it means we acquire 1200 image in real-time mode. So every 75 milliseconds we accumulate this image, which are going to show the mobility of the molecule over time. Then each of these movie, we are actually using a Gaussian fit actually to transform this big dot into a single molecule, okay, a single dot, and this is done thanks to a Gaussian field. So this is really the principle of single molecule localization microscopy, is that really on each single fluorescent probe and image, we can transform this 200 nanometer big dot into a small, tiny dot, which have a single fluorochrome resolution of 10 nanometer. So this is done automatically using MATLAB software. We have the software. The software is accessible on the internet. And so the program can reconstruct for you the trajectory of interest in yellow here. And then this trajectory, which is shown here in red, can be overlaid with a synaptic marker in white. And therefore, you can look at the mobility of your receptor outside of the synapse and inside of the synapse. And as you can see, the mobility is not the same. So for each of these trajectory, then we calculate what we call the mean square displacement function over time, which is expressed in square micrometer over time. So what does it mean? Actually, we are looking at the surface explored by the trajectory over the time of the recording. And if the relationship is linear, it means your molecule is moving further and further away. So it's not restricted in its motion. It's freely moving. And that's what we have usually for an extrasynaptic receptor. While for a synaptic receptor, the molecule is restricted in its motion. So after a while, it doesn't explore a larger surface area of the synapse or the membrane. So that's what you see here. In some cases, we also measure the time the molecule stay in the synapse. So here, this movie works. Okay, good. So you can see that the quantum dot in white without the synaptic marker, and you can see them moving along in the neuron. And you can see some that are moving a lot and some others that are more immobile. And also what you can notice on this movie is that they blink. What does it mean? It means they are losing momentaneously their fluorescence, which recover actually, okay? And this is really a particularity of the quantum dot. So it has this property to blink, which is very interesting for us because it's a way to see that you have only one fluorescent molecule at a time and not two or three. And in this case, they won't blink at the same time. All right, so a typical experiment takes, as you can see here, around an hour. So what do we do? So we first, of course, label, because this is an immunocytochemistry experiment, we label the cells. We are using, as I mentioned, these glass cover slips on which the cells are coated and have grown. And we do the labeling at 37 degrees on a heating block. To fasten the labeling. So, and here you have the condition. So for instance, we had, of course, a typical immunocytochemistry, primary antibody, we wash, we add the secondary antibody, we wash again, and then we add our quantum dots, which are coupled to, for this example, with streptavidin. But as you can see, this is very rapid. For instance, the primary antibody, you leave it only 5 minutes, 5 minutes or so for the secondary antibody, and then 1 minute for the quantum dots. And then you can mount your cover slip, mount your cover slip in the recording chamber and go on the microscope and record the movies and the synaptic image of the Jeffrey finger, for instance. So you can accumulate this image and these films from different transfected cells on your cover slip. And usually we do that for between 5 and 10 cells that are transfected. And it takes on average 30 minutes to 1 hour to acquire these 5 or 10 movies actually. Then you can store these movies and analyze them whenever you want. Because it takes some time actually to analyze. So the tracking, so what we call the tracking is a reconstruction, the Gaussian fit that is done on each molecule and accumulated over the 1200 frames. And then of course, you control that the tracking is done properly, and you can analyze the data by plotting the mean square displacement function, analyzing the diffusion coefficient, which is the speed of motion, and the explored area. So the quantum dots, you can find them in a Thermo Fisher. They come in different flavors. We like to use 655 because they are brighter. And we are using them in the coupled to streptavidin, but also in some cases coupled directly to secondary antibodies. So you can find anti-goat, rabbit, mouse, secondary antibodies. So in this case, of course, you don't have to incubate with a biotinylated secondary antibody. You can go straight and use this secondary antibody, but they are not always working. So one of the disadvantages of the quantum dot is that they can give a lot of non-specific labeling. And therefore, you have to test the two possibilities, either to use the streptavidin quantum dot or the secondary antibody to see which one is the best for your experiment. So the quantum dots, so they are nanocrystals actually. They are quite big. They are between 10 and 30 nanometer big, which is almost the size of the synaptic cleft. But they can enter the synapse, as you can see on this image. The quantum dot was revealed by electron microscopy. But so, as I said, they have the particularity, the advantage to be very photostable. So you can record a single molecule localization for up to 20 minutes. I don't know if the movie is not working also. OK. Ah, alors maintenant je suis sortie. OK. I think. Okay, no, so movie are not working. Okay. So here, for instance, it's a reconstruction of a trajectory over the 20 minutes. So you can see that the receptor he went on these dendrites, along this dendrite, and explored all these dendrites, this dendritic bifurcation actually for extrasynaptic receptor, while at the synapse, it actually stayed at the synapse for the 20-minute recording. Okay, so you cannot see it here, unfortunately, but when we use organic dyes like size 3, we can absolutely not see such a long recording. Most of the time, the recording lasts for 5 or 10 seconds. So it's because the organic dyes are bleaching very rapidly, and so they lose their fluorescence, and these fluorescence don't come back anymore. All right, so this was to show you also that so to show you that actually if you label a molecule that is not supposed to be enriched in the synapse like the receptors, or for instance an adhesion molecule, NCAM here, you could see, but you can see it actually on the trajectory, that it explores the synapse the same way than the extrasynaptic membrane. So it's not accumulated at the synapse like you can see here for this synaptic trajectory, where they spend much more time at the synapse than here, where you see a molecule that cross actually several synapses during the 20 minutes recording. Okay, so yes, so using this technique, we could actually image the neurons for up to 1 hour and look at the localization of the receptor during the 1 hour recording. And as you can see, some receptors, so first of all, you can see that on the same neuron, you have different behavior. You have receptors that stay at the synapse during the whole recording. You have some that enter the synapse, some others that exit the synapse, some others that can transit between synapses. So each individual receptor may have different behavior. So that's one important thing to know. And as I mentioned you before, we noticed also that from the MSD and from the calculation that the synaptic molecules, they are usually confined at the synapse. They are slowed down at the synapse. If you look at the cumulative probability of the diffusion coefficient, This is shifted to the left, meaning towards lower values, meaning the receptor is slower within the synapse and more confined. And so this is really, you can find that for GABA receptor, glutamate receptor, P2X receptor, whatever receptor you want to look out. It's a general mechanism that happens for receptors, synaptic receptors. Okay, so just for troubleshooting of this technique, so most of the time the main problem we have is based on the poor or the excessive quantum dot labeling on the neurons. So you can have too much quantum dots. If you have too many quantum dots, then all the trajectory, reconstructed trajectory will cross over, and then you won't be able really to analyze the diffusion of a given receptor. So it's going to be very complicated for you actually to analyze this data and also to extract the result. Of course, the opposite is also a problem. If you have a poor quantum dot labeling, knowing that the behavior of the receptor is really changing depending on the synapse you are looking at, depending on the receptor you are looking at also, you need a lot of quantum dots to have a population study to really extract a median behavior of the mobility. So most of the time, the main problem is the primary antibody. So you need to have a very good antibody that works very well for your regular immunocytochemistry on fixed cell. And of course, it has to be an antibody that recognizes an extracellular epitope of the molecule. And so you can play on this antibody concentration and time of incubation, but it shouldn't be more than 15 minutes incubation. Otherwise, you have non-specific labeling. Also, you can have non-specific labeling with an excessive, of course, quantum dot concentration. And as I said, these are crystal, they are sticky, and they can give a lot of background. So you have to be really careful to use it no more than 2 nanomolar concentration and for a very short period of time, so 1 minute or 2 minutes maximum, not more. And also the last problem you can have is an excessive blinking. So the quantum dots, they blink, it's good, but if you shine too much light, they will blink too much. So you have to, and excessive blinking you will have very short trajectory at the end and not very good precision on the trajectory. So it's just to shut a little bit down your light. Okay. So I try to be fast. So now I'm going to talk about, I will show you examples later on of single particle tracking with quantum dots and what we can do also together with the STORM Palm super resolution. So now I'm going to talk about STORM Palm super resolution. So STORM for stochastic optical reconstruction microscopy and PALM for photoactivated localization microscopy. So actually this is the same technique. So it's single molecule localization microscopy. We have to do a Gaussian fit also on single molecule, but on fixed cells. And this is very convenient. So this is our setup in the lab. And this is an important method that obtained actually the Nobel Prize in 2014, and which is really helping the researcher to look at the organization of molecules within very tiny environment like the synapse. As I mentioned in my introduction, that's the only possibility to see nanodomains in the synapse, because these nanodomains, they are between 50 nanometer and 100 nanometer. So we are, in this case, below what we call the barrier of the diffraction of the light, which is due to the fact that the light is a wave, and therefore all the fluorescence microscopy won't be able to show you really a structure that is below the size of 200 nanometers. So to go below 200 nanometers, so to see the nanodomain, you need to go to this technique. So this technique, as I said, is also a single molecule localization microscopy approach. But in this case, we are using fixed sample, and we are actually activating single molecule localization randomly on the sample, and we are doing what we call a pointy stick image. So we reconstruct the image point by point after turning on a single molecule on and off, so you can do the Gaussian fit for individual molecules, which prevents you to have a big dot like that when the two molecules are too close to one another. Okay, so you can resolve individual molecules much better than that. So this is the idea. So indeed, so this is typical fluorescence. You shine light, you excite all the American fluorescent molecule at the same time. But for super resolution, you control the activation of your single molecule. They are activated, and then you can also turn them off. So you turn them on and off using different lasers, and you can reconstruct in black here, by Gaussian fit, the localization of individual molecule, and at the end you have a super resolved image, pointy stick image, using again the Gaussian fit. So it's what it's shown here for the Eiffel Tower. You see it shining, and then we can reconstruct one by one the localization of this single light, and have a very good reconstruction of the Eiffel Tower. All right, so that's what we used here, for instance, to look at the localization of a receptor, which is not the GABA receptor, it's the adenosine receptor. In green here, you see Jeffrin. So you see the synaptic scaffold for the GABAergic synapse. And as you can see, this receptor is not within the scaffold, but it's around the scaffold. Okay, so as you can see, these particles of receptors, these mini clusters, they are all around the synapse. So that's how we could say that this receptor is actually perisynaptic. It's not inside the synapse. And using many other techniques, we came out with a role of this receptor in the formation of the synapse by having a role in the stabilization of the synapse. We also use this technique. For instance, this is a typical dendrite of a neuron. This is palm microscopy, actually. And you can see in green the conventional fluorescence image, and you can see the super resolved image of the same dendrite, and now you can see the nanodomain of receptor that you couldn't see before using a palm microscopy. Okay, so palm microscopy or storm microscopy, you cannot use the quantum dot. It doesn't work. So that's bad because the quantum dots are super bright, as I told you. So we need to use some other fluorescent probe, and these probes, they are very dim. So we need again to increase the signal to noise ratio to have a good signal, to have a good Gaussian fit. So it's the same principles, and for the quantum dot SPT, we use an inverted microscope. And we also use very good objective, and this time we use an objective that is really dedicated for super resolution. Most of the time it's a 100x objective with the highest numerical aperture, so the highest opening of the lens, so you can collect a maximum of photon, and we use a 1.49 numerical aperture. So such an objective, and also these objectives are very peculiar because they are corrected for aberration. So you have no aberration in the light, so you can collect a maximum of light. So they are super expensive, and they are really dedicated for this experiment. We also use EMCCD, so it's the same principle. That's what you see here. The camera that is very sensitive and that is cooled, so you're at -60 degrees, so you can have lower background. And this time we need lasers to turn on and off our single molecules. We cannot use the lamp because for the fluorescence image. And to decrease also the background, to increase the signal-to-noise ratio, we are doing what we call turf illumination, which means that the laser is tilted, so you can light up the samples only on the 10 micrometer in contact with a glass cover slip. So you prevent to have the background from the top of the image. All right. So the difference between the Palm and the Storm super resolution is based on the dye that you are using. So for the Storm image, we are using synthetic dye, organic dyes like Alexa dyes, CY3, CY2, CY5, the typical synthetic dyes. While for Palm, we are using fluorescent protein. And most of the time, this is like a GFP protein that can convert between green and red. So that's the photo activation. So that explains why it's called photo activated laser microscopy, localization microscopy, sorry. And so the big difference between the two is that you need to really to transfect your cell with for instance, the subunit of the receptor, for instance, gamma-2 subunit of the receptor that is coupled to this GFP protein. So we are using most of the time Dendra2 or MEOS, which are the main photoactivatable protein. While for the Storm, you can actually use an antibody that is directly coupled to the dye, and therefore you can look at the endogenous receptor. So that's an advantage, but you can also, of course, look at some recombinant proteins. With the two techniques, you can do live and fixed sample, and you can have also multicolor. But you will always have a better resolution for the Storm than for the Palm because the organic dyes are brighter than the photoactivatable molecules. So it's maybe complicated to understand, but okay, so to turn on and off our molecules, we are using a combination of lasers, and especially the UV laser. And so this is really based on the principle, physical principle of fluorescence. So when you excite a fluorescent probe, it emits, it goes on state, it emits some light, and then it goes back to what we call the ground state to be excited again. Okay. And before to go back to this ground state, it goes into what we call a triplet or dark state. Where this is an off state. So the molecule is not fluorescent anymore, but it can go back to the fluorescent mode if you shine UV light and you help it to go back to the excitation. So that's really the principle how we turn on and off these single molecules. We can synchronize and activate them using UV light. So to control the off state, so you want to keep them a little bit longer in the off state, we use what we call a scavenger system that remove oxygen from the buffer to keep this off state much longer. So this is a typical buffer that we need to add to our cover slips before imaging to help the fluorescent dyes to go into the dark state and to be activatable at one point using the laser. So here is a typical example of an experiment using Storm. So you have again, immunocytochemistry for endogenous receptor, primary, secondary antibody with a fluorescent dye and with the and so we accumulate also between 20 and 30,000 frames with an acquisition time of 100 milliseconds. And you can see these single molecules that get randomly activated. And we do that on each frame. Then we are going to reconstruct with a Gaussian fit the localization of the molecule. And we get this pointy stick image after reconstruction that we can quantify using specific program. I don't have the time to discuss this program. They are accessible actually freely. We have one that we are using and that we actually modify, so it's really good to be able to see the cluster and to resolve the cluster. But you also have some plugin on ImageJ that exist and that you can use. Okay, so we are actually right now writing a protocol for the single molecule localization study of the GABA receptor. So if you are interested, it's not published yet, it's ongoing. And in this protocol, we are also showing the timing of the event. So it's a little bit longer than the quantum dot-based single particle tracking. It takes 3 hours, mostly the experiment. So again, so primary antibody incubation, secondary. So the incubation is a bit longer than for the SPT. And then you mount your sample once labeled in the recording chamber, and you add what we call the storm buffer, so with a scavenger of oxygen inside. And just before imaging your cells, you need to add a bead, a fluorescent bead, which is of size of 100 nanometer. And as you can see here on this image, after a reconstruction of your image, these beads are actually very useful to help to correct the drift that can happen during the acquisition of the 30,000 frames, because it takes a lot of time to acquire these. It takes several tens of minutes. So most of the time we have a drift in the image. So you can correct this drift thanks to these beads. Okay, so for storm imaging, the best antibody and fluorescent probe that we are using is an Alexa 647 because it's very bright and it blinks also very easily, and you can keep the fluorescence a long time also. And you can turn it on with a 637 nanometer laser. And sometimes you need to push the blinking with a 4 or 5 nanometer laser. I don't want to go in details about that, and we acquire using most of the time 30,000 frames to get a very nicely resolved image. With Palm, of course, you don't need to do the labeling. Primary antibody, secondary antibody, you look directly at your sample that has been transfected with the photoactivatable GFP, and you just turn it on with your laser. So you go from, the molecule will go from green to red point by point also in your image. And most of the time we do 20,000 frames imaging. All right, so here is a typical dendrite image. We take a picture with white field, so standard fluorescence to look at the region of interest, and then we acquire one by one the localization of the single molecules for 20,000 frames. We reconstruct using MATLAB also software with a Gaussian fit the pointy stick image. We correct the drift, okay, on this image, and then we can now have access to the nanodomains and to the organization of the receptor within the synapse. And so we quantify this localization using our by thresholding actually the cluster that we want to analyze. And in this cluster, we can have access now to the molecules that are present within the cluster. So you can have access to the size of this cluster and to their structure. And of course, the amount of receptors that can be inside. So this is just to show you the different, the comparison between the different super resolution techniques that exist. So the Palm and the Storm are the most, as you can see, are the ones that are have the best resolution in terms of spatial resolution. So you can resolve the localization of molecules that are spaced by 10 to 40 nanometer. And you can also have access on this table to the different advantages and disadvantages of the different technique. I'm not going to go into details, of course. Okay, so just to now show you what we can do and what you can, how it can be useful also for drug, I mean, if you want to look at impact of some drugs on your synapse. So as I mentioned before, so using this technique, we could show the 2 nanodomains of GABA receptor that are present at the synapse, as you can see here. You can look at them in 2D and also in 3D. Okay. And you can see that this is very different between the receptor and the Jeffrey scaffold, which is quite homogeneous. So you have a homogeneous Jeffrey scaffold, but you have nanodomains of receptor, and you can quantify this data by measuring the number of these domains. So as I said, you have 2 domains for the receptor in the synapse, while you have only 1 for the Jeffrey scaffold, as well as you can characterize their size, and of course the density of molecules you can find within the synapse, which gives you an idea of the number of molecules within the synapse. So using single particle tracking, we quantum dot-based single particle tracking, we studied, for instance, the impact of a human mutation, okay, that is associated with the GABA receptor, and we found that a receptor that have this mutation is actually more mobile and it escapes faster the synapse. And we connected this mobility with a reduced efficacy of the synapse, as you can see here from these mini iPSC's recordings. We also used this technique to study the effect of benzodiazepine, which are allosteric modulators and which are anxiolytic molecules, very famous anxiolytic molecule like Xanax or whatever. And you have different flavors of this benzodiazepine. And as you can see, a particular one actually increase the mobility of the receptor that escape faster the synapse. So that's what we showed. So we showed that the benzodiazepine, the first effect of benzodiazepine is to change the mobilization of the receptor, GABA A receptor at the synapse. And now, so what we are doing right now in the lab is to use these different techniques to look at drug design. So very, very rapidly. So as I mentioned, it's important to look at the mobility and the nanodomain organization to understand the mechanism of regulation of receptor at the synapse. But you can also change the efficacy of the synapse independently of the receptor, for instance, by looking at some chloride transporter. So why? Because the receptor is permeable to chloride, this GABA receptor. So chloride can go in or out of the receptor. And this really depends on this intracellular chloride level, which is really dependent on these two transporters, NKCC1 and KCC2, which are important for the influx or the outflow of chloride from the neuron. And we know that at mature stage, you have a low chloride, and therefore the GABA receptor, once it is opened by a GABA binding to the receptor, can make the chloride goes in, and this is responsible for the inhibitory action of GABA in the neuron. But while at immature stage, you don't have this KCC2 transporter, you have NKCC1, and you have a big influx of chloride in the neuron. And therefore, the GABA receptor is going to exit chloride from the neuron. Okay. And in this case, sorry, the GABA is depolarizing or excitatory. Okay, sorry about that. So, but we know, oh là là, j'ai plus de voix. We know that in the pathology, actually, you can go back to an immature stage by losing KCC2 at the surface of the neuron. So KCC2 is really unstable in the pathology. It can be internalized and degraded. So in the pathology, like epilepsy, chronic stress, psychiatric disease, we are searching for molecules that can actually stabilize KCC2 in the membrane. So that's what we are doing in the team. And we showed that these transporters, they are perisynaptic actually, and they form this aggregate also, this cluster, and they respond to the diffusion mechanism that we found for the receptor. As you can see here, they diffuse fast outside of the synapse. They are confined near the synapse, but they can escape the synapse. And this is also very rapidly regulated by activity. And we are now focusing on the regulation by GABA, a receptor itself of this transporter. And so when we found that actually GABA regulates this transporter very rapidly by chloride itself. So the chloride that goes through the receptor, thanks to a kinase that is not very well known, which is called the Wnt kinase, that can be activated by phosphorylation when the chloride is very low in the neurons. And in the lab, we showed that this kinase, once activated, it activates a second kinase, which is called SPAC. And SPAC is going to phosphorylate all the molecules that are very important at the inhibitory synapse. So KCC2, NKCC1, and gefferin, and which controls the synaptic localization of the GABA receptor. So we believe this kinase is actually a new target, could be a new target in the pathology. And we showed that in epilepsy, for instance, you have a huge activation of this kinase. So the aim is to block this kinase to prevent the internalization, the endocytosis of KCC2, and therefore the increase in the intracellular chloride and the depolarizing excitatory action of GABA in these neurons. So we are searching for a new mechanism of regulation of this KCC2 through this kinase. And Erwan in the lab, which is a PhD student, is looking at a mechanism by the way. He noticed that NKCC1 has two SPAC binding sites that are shown here, while KCC2 has none. So we propose that NKCC1 actually regulate KCC2 by recruiting SPAC to the membrane. So this is what is schematized here. Okay, NKCC1 and KCC2, by being close enough, NKCC1 will recruit this kinase that will regulate phosphorylation of KCC2. So if we block with recruitment of SPAC on KCC1, we should block the regulation of internalization of KCC2. So Erwan showed that this is probably indeed working this way by looking first at the colocalization of the two transporters. And as you can see, they colocalize pretty well at the surface of the neuron. And using storm palm imaging, we could show that the transporters, KCC2 and NKCC1, they colocalize at the periphery of the synapse. So that's how we represent them. And using single particle tracking, it could also show that KCC2 is spending a lot of time, more time actually, in an NKCC1 cluster, and it's also slowed down and confined within this cluster, but it can escape this cluster and goes faster in the membrane. Okay, so based on this result, we propose that indeed they are very close to one another, and they probably interact together because they have this particular relationship of KCC2 diffusion once it encounter NKCC1 cluster. And when we quantify this mobility, you can see indeed that KCC2 on NKCC1 cluster is much slower than KCC2 outside of this cluster. Okay, so we believe we have a new mechanism of regulation of KCC2 by NKCC1 by recruiting the spike kinase. So what Erwan did is to actually design peptides that mimic this spike binding site on NKCC1. So these peptides, they can block the access of SPAC to NKCC1, and therefore the phosphorylation of KCC2 on these two threonine key sites and its internalization. So by raising tens of peptides, he found actually one in blue here that is really a good could be really a good tool because it's increasing the clustering of KCC2 at the surface of the neurons, while the others are either decreasing or having no effect. So there is this peptide 5, that we tested now in vivo. So first of all, yes, I want to show you that this peptide that is in red here is actually, by modeling, you can see that it's very well positioned to prevent the SPAC in green here and NKCC1 interaction. So indeed, it makes sense also with the effect we saw on these neurons. And the last thing Erwan and Celia, a postdoc in the lab, did is to use this peptide in vivo, because the idea is to develop these tools to prevent KCC2 internalization and therefore GABA depolarization and excitatory hyperactivity in the brain. So we induced epilepsy, acute epilepsy in these animals, and Erwan and Celia looked at the effect of their peptide on these acute seizures, and as you can see, it decreased significantly the duration of the seizure, the acute seizure, and then on these animals, and also decrease the deaths that can happen after a seizure in these animals. So we believe this peptide is very promising for the regulation. Okay, so I'm done. I just want to show you a picture, so it's not an up-to-date picture of the team with the different people of the team that are working with me. I don't have the time to go through all this person, but and also our collaborator and our funding, and of course I thank you for your attention.